Now that the coronavirus has reached Ireland, we have been struck by a much worse epidemic: misinformation. When it comes to science, you can get information from a number of sources, but the best bet for accuracy is in scientific journals and papers, which describe studies and experiments and have been peer-reviewed by other scientists. These papers can be difficult to understand, expensive to access, and hard to find, so I've spent some time reading the ones I think are relevant and condensing what they say into this video. Presenting COVID-19 Explained by an undergraduate science student. So to talk about COVID-19, I'm going to break it down into a number of steps to make this a bit easier to follow. These steps are origins, spread, prevention, testing, and treatment. The first cluster of COVID-19 cases was reported as unusual pneumonia by clinicians from the Wuhan province in China to the WHO on December 31st, 2019. That purple star marks a reference. I'll be leaving them all over this video so that the papers I'm talking about are easily identifiable. In less than 4 weeks, the virus had been identified, its genome sequenced, and a test developed, which we will talk about later. The cause of these pneumonia cases was identified as a novel member of the coronavirus family, now called COVID-19. Coronaviruses are a large family of viruses with an RNA genome, different from the human DNA, surrounded by a lipid envelope that looks like a crown, hence corona. The envelope is covered in spike proteins, which is something to remember for later on when I talk about treatments. Members of this family include common colds, but also include more serious illnesses, such as MERS and SARS, with SARS causing a similar but less far-reaching outbreak than this one back in 2003. But where exactly did COVID-19 come from? The short answer is we don't really know yet. Researchers will likely use phylogenetic analysis, which is basically using similarity between genetic sequences to construct a kind of family tree, to develop theories about where the virus may have came from. However, no matter where it came from, we know it's basically going global, so let's talk about why. It's time to talk about how the virus spreads and why it's gotten as far as it has in such a short space of time. During an epidemic, or in this case a pandemic, there is one important number known as R, or the reproduction number. This is the approximate number of people that will get infected by one infected person. If it's more than one, an outbreak is sustainable and will continue. This number is based on different factors and changes all the time, but R0, or the initial transmissibility of COVID-19, was estimated to be approximately 3. At present, the virus is believed to spread from person to person via droplets or aerosols from their respiratory tract. When expelled from the body, viral particles remain viable or ready to infect for approximately 3 hours. Put that together with the fact that the range of the average adult cough is about 2 meters, and we start to understand why this virus is spreading so readily. Now, that's direct person-to-person transmission, but what about commonly touched surfaces like tabletops or shopping trolleys? The best evidence I can find as of March 15th, which is based on a controlled scientific study, suggests that the virus can survive for 24 hours on cardboard and up to even 72 hours on plastic and stainless steel. When exposed to the virus, there is an incubation period during which you don't show symptoms. Important to note, however, is during this time you are still infectious, possibly even more so than after you become symptomatic. As of March 13th, this incubation time from exposure to symptoms is 5 days. 97% of patients will show symptoms by 11 days, and 99.99% of patients show symptoms by 14 days. This is why later, when we talk about quarantine and isolation, the magic number is 14 days. We know how it spreads now, and we know that it's spreading fast. So before I go any further, I'm going to talk about prevention. First of all, let's talk hygiene. Why all the hysteria about hand washing? Well, soap is full of fatty acids, which break down the lipid envelope of the virus and essentially kill it. The same is true for hand sanitizer, assuming the alcohol content is high enough. The reason it's important to keep your hands clean is, as we have seen, the virus can survive on surfaces for long periods of time. Your hands come into contact with many of these surfaces, and in turn will touch your face. Next, let's talk about a concept most of us had never heard of until this year, but now appears to be one of the most discussed ways of preventing the spread of coronavirus: social distancing. Most people know how social distancing works, so I won't go into detail, but I do want to talk about what happens if it isn't done right. Social distancing doesn't have to be a solitary activity. You can quarantine yourself with your family, friends. Isolation cells of a small enough number is fine. The only caveat is that everyone in your isolation cell must only come into contact with other members of your cell, and nobody else. If one person interacts outside of the cell, your whole unit is compromised. So if everyone was to sort themselves into cells and isolate for 14 days, some cells will have an infected person and others inside the cell would become infected. Cells that were uncompromised will know after 14 days, if none of them show symptoms, that they are coronavirus free. Then they can merge with other unaffected cells, provided those cells have also been uncompromised for 14 days. However, if during this 14-day period, even one member of each cell interacts with a member of another cell, the virus will be able to travel between isolation groups and we are back to a level of unknown about who is infected and who isn't. I also came across an interesting study to say that athletes may be greater spreaders of respiratory illnesses, due to enhanced lung performance expelling pathogens from their respiratory tract with more force. So maybe that's a good excuse to avoid the gym for the next while, that is, if it's even still open. So, if you've been exposed to COVID-19 and you are showing symptoms, what's next? Before I go on, I'm going to remind you that I'm just talking about the science here, so I'm giving no information on where to go to get tested. What I will talk about is the scientific basis behind the test. As I mentioned, the WHO was very quick to respond when this virus was identified in December, and by January 26th, a test had been developed. This test is called a real-time reverse transcription polymerase chain reaction test, or real-time RT-PCR. Earlier I made a point of mentioning that the coronavirus family has an RNA genome. It's a well-known fact that we all have genetic material that contains the instructions for who we are, and in humans that genetic material is called DNA. In human cells, DNA is actually a blueprint our body uses to make a different molecule called RNA, which in turn makes us who we are. COVID-19 starts with this RNA as its blueprint. Just like our genetic information is unique to us, COVID-19's RNA is unique to COVID-19, which means if we want to look for its presence, it's a good idea to look for its RNA in the respiratory tract of patients. A sample is taken from this area and the RNA extracted from the microbes present near these cells. This RNA is converted back, hence reverse transcription, into DNA, and mixed with two components, primers and probes. The primers have been specifically designed to identify and bind to specific sequences in the COVID-19 cDNA and cause it to be amplified, or multiplied, in cycles, in machines called PCR machines. If viral cDNA is present, it will be amplified with every cycle, and this action causes the probe to be broken down and to fluoresce. The fluorescence intensity is measured by a PCR machine, and if a sample begins to fluoresce sufficiently, that sample is positive for COVID-19 cDNA. Therefore, it contained COVID-19 RNA, and therefore it contained COVID-19. To explain why shortages of testing are happening in some countries, I want to draw attention to the primers. The primers are specifically made for this test, and shortages of primers mean that the test can't be run. Okay, we have covered origins, spread, prevention, and testing. But for many patients, unfortunately, their worst fears will be realised, and their test will come back positive. Let's talk about what that means. I'm not a doctor, and as such will be giving absolutely no advice on what drugs to take. But I am going to discuss what the literature says about what drugs are in development and how effective they have been. For kids, it seems like even when infected, healthy children are left relatively unscathed by COVID-19. We are still largely in the dark as to why this is the case, but it may be linked to the receptor the virus uses to interact with and enter our cells. Every cell in our body is covered with receptors that cells use to interact with each other and extraneous objects. COVID-19 has been shown to enter our cells via interaction with the ACE2 receptor, with the COVID-19 spike protein interacting with the receptor. One explanation for children's apparent immunity to COVID-19 is that their ACE2 receptors may not be fully formed or have a different structure. Designing a new drug de novo, or from scratch, can be costly, error-laden, and time-consuming. That is why at times like this, researchers try out drugs that have been proven to work with other illnesses, and see if they can make them work for the problem at hand, in this case COVID-19. There are some trials that are repurposing drugs from other illnesses and do seem to be having an effect. One caveat of this is that these drugs were not originally designed for COVID-19. As I mentioned, ACE2 receptors are the method by which COVID-19 enters our cells, so treatments that inhibit these receptors should be promising. COVID-19 binding to ACE2 also utilizes another molecule called TMPRSS2. A drug called camostat mesylate, which has been approved in Japan for clinical use against pancreatitis, was shown to inhibit the action of TMPRSS2 and therefore the entry of COVID-19 to cells. However, repurposing of drugs to fit this new purpose will not always work. For example, one effect that COVID-19 can have on patients is the initiation of a cytokine storm. This is an autoimmune reaction which causes out-of-control inflammation and eventual organ failure. In China, doctors attempted to use corticosteroids to treat this reaction. If you don't recognize corticosteroids, you might know them from the brown inhaler used to treat asthma. However, use of corticosteroids against COVID-19 in this case was not found to help, and in fact delayed clearance of the virus. Remdesivir is an anti-RNA viral drug which was undergoing clinical development for treatment of the Ebola virus, and has been theorized to also be a candidate for treatment of SARS-MERS, but hasn't been tested yet for this in humans. Lab testing of cell lines treated by remdesivir has shown that it may be able to inhibit and clear COVID-19 infection. Clinical trials are now taking place using this drug to treat COVID-19, and I know of one patient in the US who did clear the virus when treated by remdesivir. That same study also examined the use of a drug called chloroquine against COVID-19. Chloroquine is an anti-malarial that is generally regarded as very clinically safe and has been used in humans. The results of that study are quite promising, especially as chloroquine is so widely used already. I don't have a reference star drawn for this because this is a paper I only found quite recently, but 2 days ago a preprint was published of a study in France where 20 patients who were treated with a combination of hydroxychloroquine and azithromycin showed clearance of COVID-19 within 6 days. Azithromycin is a very common drug used to treat respiratory infections such as strep throat. Finally, the first drug approved by China as an official anti-COVID-19 drug is something called favipiravir. Apparently, the drug has shown efficacy at treating patients in a clinical trial with a sample size of 70. I know people are interested in finding out about vaccines, but it could be many months before we have anything more than speculation to talk about. But why do vaccines take so much longer to develop than the drugs I've already mentioned? Therapeutics which are delivered to sick patients can be approved quicker. There's a higher benefit to risk, as they will only be administered to patients who are already sick. Furthermore, the population of people who will be taking those drugs are lower, as it only includes infected people. Vaccines, on the other hand, are designed for distribution to the whole healthy population. The risk and variation within the whole population is far greater, which is why vaccines require much more intense testing. That's what the science is saying about COVID-19. If you've watched this far, I hope you've enjoyed it, and this was helpful in any way. In the coming weeks, pay attention to the sources you're getting your information from, and be safe. If we will learn anything from this virus, it's that the borders between our countries mean nothing. We all live in different countries, but essentially we all live in the same place, on the same planet. What affects one of us affects us all. I'd like to thank my classmates John Fehley and Emer Wall for helping me put this video together. And UCC, if you want to just give us credit for this video and let us graduate, that would be class.